Hi, everybody. Welcome to Micro. So what I'm going to do for you today is I'm going to record myself performing a streak for isolation lab. With this lab, I've already have bacteria that's prepared. So we did this first semester virtually. I'm going to streak from one Petri dish to another Petri dish. Okay. So with Micro, even before COVID, we had sterilization procedures in place because we're working with bacteria. So I'm going to model those for you here as well. So every time before we do a lab in Micro, we have always washed our hands and sterilized our station. We do it before each lab and at the end of each lab. So that's what I'm going to go ahead and do. So I'm going to wash my hands. Okay. And we go through tons and tons and tons of paper towel in Micro in a normal year. Okay, we have garbage cans underneath each of the lab benches. And then I'm going to go ahead and spray down my station. Now, one of the precautions we are also taking this year with COVID is that we are going to wear gloves because we're all handling and sharing instrumentation. Okay, and so just as a precaution, we're going to wear gloves while also doing micro labs this year. In a normal year, that is not a precaution we typically take. What we would do is if you have any cuts or open sores on your hands, you would wear a Band-Aid to cover that up because as Ms. Smith and I like to say, if there's a hole, it can get in. So we're working with bacteria. We're going to be extra cautious. Okay, so we have to use, I have to light a Bunsen burner. We have to use a Bunsen burner for this lab. So for those of you who don't remember from chemistry, perpendicular is off, parallel is on. So I am going to turn my gas on. I already turned on the main supply. And there's a slight hissing, so I know that the gas is on. I'm going to open the valve down below. I also have my hair pulled back, so that way, just to make certain that we're all safe. So this is my flame. I need to adjust my flame and turn it down a little bit. It's a little big. Okay, so lefty is loosey for bigger, righty is tighty. So I'm going to adjust my flame. Okay, there we go. There we go. I was going the wrong way. Okay, my bad. Okay, so what I am going to do, I have some different Petri dishes here of bacteria that's already been streaked. I have, you can see that Serratia Marcescens, abbreviated S mark. The date I streaked has been rubbed off, but you can see it on this one, the B Meg that I streaked. They were streaked on 3/12, so it just got rubbed off on this one. It was labeled. We always label our bacteria with the type and the date that it was streaked. So I'm going to go ahead and I'm going to use S mark today. That's the bacteria I'm going to use. I like that I can see it very easily. And on my Petri dish, my new Petri dish is blank. I'm going to break this up into 4 quadrants. I don't know if you remember this from the online lab that you did, but I'm going to label them A, B, C, and D, like that. Okay. And then I'm also going to label it with my name, the one and only Stoles. Okay. S mark, that's the bacteria I'm streaking. And then today's date is 3/18. I'm going to put that on the Petri dish as well. Okay. So we are all labeled. So that way I know which Petri dish is mine when I go and check it in a few days. Now, these Petri dishes, when I grabbed them off the lab bench, they were upside down. We always store Petri dishes upside down. So that way any condensation that might forms on the lid of the Petri dish and not on any bacteria that we are growing. Okay. And we always label the bottoms of the Petri dishes, not the lids, because sometimes the lid becomes separated. And we need to be able to know what is on the Petri dish or what's being grown in the Petri dish. And if we label the bottom, it doesn't matter if the lid becomes separated because the label is still there. Okay, so what I am going to use today, I'm going to turn these back over, my Petri dishes. What I'm going to use today is my inoculating loop. So here is my inoculating loop. It's got a little loop on the end, okay? This is the tool or instrument that I use to transfer bacteria from one Petri dish to another. Okay, but I have to sterilize it first. It's been sitting out, it's been used, it's transferred other bacteria in the past. So I am going to sterilize it, okay, so that way it is good to go. Okay, normally, about 30 seconds roughly. You can always do more. Okay. Now I need to let it cool. It's really important that I'm letting this cool because I'm trying to transfer bacteria. I want the bacteria that I am transferring from the initial Petri dish to my new Petri dish, okay, to survive. I'm trying to get them to grow. And so I am going to let it cool. I'm not going to place it down to cool back on the lab bench because if I do that, it's now I need to sterilize it again. I'm not going to blow on it to cool it off either because the bacteria from my mouth will then contaminate it. I must hold it and I just wait roughly about a minute for it to cool. So now what I'm going to do is I'm going to get let me tilt my camera down so we can see, my S mark. And I'm going to open this Petri dish, but I'm going to open it Pac-Man style. I'm not going to take the lid off and put it open on the desk, on the lab bench, because there's a risk of more contamination. So we're going to do it Pac-Man style. So I'm going to open it very gently, just Pac-Man style. Okay, and I'm going to just very gently grab my bacteria off the Petri dish. And I don't know if we can see, but I have my bacteria right there. Okay, I am now done with the original Petri dish, so I'm going to put it right there upside down. Now in my new Petri dish that's already labeled, I am going to open this Pac-Man style and I'm going to streak it in quadrant A and just quadrant A. So here we go, back and forth in quadrant A. Now the agar it can be delicate. You just have to do gently on the top. You don't want to gouge the agar, okay? Now I'm going to sterilize my inoculating loop again, okay? Sterilizing it again, I need to kill off any of the excess bacteria that did not transfer from my original Petri dish to the one I'm trying to do a streak for isolation in. Okay, so now I'm going to wait. I got to let it cool again because once again, I am trying to keep my bacteria alive. I'm not killing them. Okay, so roughly a minute. Do, 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 do. And for Miss Smith students watching this is my personality all the time. Okay, so this should be cool now. I have my bacteria that I streaked in quadrant A. What I'm now going to do is I am going to transfer from A to B. So I'm going to open this Pac-Man style again, and I'm going to grab from quadrant A and pull into quadrant B, and then stay in quadrant B and go back and forth. I'm trying to thin out my sample so that by the time I get to quadrant D, the goal is to see individual colonies of bacteria. Okay, that's the goal. So I'm going to go ahead and open Pac-Man style and pull very gently from A to B. Okay, and what I like about the S mark is that I can see it. I don't know how well you can see that on your camera. So I know I can tell if I've pulled the bacteria. Some bacteria is a little harder to see, so you may not know. I am now going to sterilize my inoculating loop again. Okay. And then once again, I am going to wait for it to cool. So once as I'm done with all of this, the bacteria sample is going to go in the incubator. The incubator is in my back office area. It is kept at roughly 37 degrees Celsius, optimal growing temperatures for our bacteria. What we're growing this on is nutrient agar. Okay, nutrient agar. There are different types of agar that exist, and they're used for different test purposes, etc. Nutrient agar, it's pretty much just the standard. Think of it as like the control upon which all bacteria is grown. Okay, so now I'm going to, it should be cool. I'm going to pull from quadrant B to quadrant C. So starting in B, pull through to C and thin out. Okay, that's it. I am now going to sterilize my inoculating loop one more time. We're almost done. Okay, let it cool. And so then we just have to go from C to D. This procedure, if it was a normal school year, by the time, I don't know, by the time we do procedures more than once in micro. It's very rare that we would do a lab just one time. So you guys would normally perform a streak for isolation at least 2 or 3 times in a normal school year. It becomes second nature. It's one of those things the students really don't even have to look up the directions for the procedure to know what to do and to go from there. So those of you who are not coming to school, at least you're seeing the video of it. I don't know why I just put that back in the flame. I wasn't thinking. I got to let it cool some more. And at least you've seen a video. So if you are then going to do these in college, you have some idea as to what's going on and what the purpose is. Okay, so I'm going to go ahead one last time. I'm going to go from C to D, Pac-Man style. Pull from C into D. And sometimes when you streak, you may not see any bacteria in the quadrants. That doesn't mean that nothing is there, nothing's growing. It only takes one bacteria to be able to replicate. So I sterilize my inoculating loop one last time. I now place it down on the lab bench because I'm done with it. Let me turn my camera back up. Okay, and now I'm going to turn off my gas supply. So we are done with the gas. We do not need it anymore. I'm going to turn it off. We do not blow out Bunsen burners. They are not birthday candles. So I'm going to turn it off back to perpendicular. All good. Okay. What I am going to do, I'm done with my gloves. I'm going to take them off. I am going to wash and sterilize my station, wash my hands and sterilize my station one last time. And then I will be placing my bacteria into the incubator, to see how it grows and what it would look like. So once again, I did Serratia marcescens, S-Mark. So for those of you at home, you should be looking up an image of what a streak for isolation of S-Mark would look like on nutrient agar. Okay, so you kind of know what you should be expecting to see or what you would be expecting to see if you were to be in school and be performing this lab. So that would be part of your assignment. Look that up. What would a streak for isolation look like of the bacteria Serratia marcescens? Okay, also abbreviated S-Mark. What it would look like on nutrient agar. So that's it. That's streak for isolation. We don't get the results right away. Okay, it would be at least 24 to 48 hours to see what our bacteria is going to look like. So those of you at school, you'll just get to see them the next week. And that's it. So yeah, enjoy.